right? So once done, like, let me know. Over.
Yes, sir.
Okay. So now we will talk about, first of all, let's talk about stereo system. Now, what is the meaning of stele? The meaning of stele is that is a vascular bundle plus pericycle. Okay, that is actually called as a stele, right? So just now we have discussed that we can sew the vascular bundle of xylem like, pteridophyte like this. So this structure is a xylem, and this structure is a phloem. Okay. Now vascular bundle can be of three types in pteridophyte. So what are those? The first, what we call protostele. The second, what we call syphnostele. And third, what we call solenostele. Okay. Now, what is protostele? So the diagram which right now I have drawn, it is a protostele, right? Now, depending upon the different shape of xylem, different types of protostele has been recognized. Okay, what we can say that on the basis of structure of xylem, different types are recognized. Okay, so what is the first type? The first type we say haplostele. Now, what is haplostele? The diagram which I have drawn is a haplostele. What we can say? Xylem is simple. Okay. Second, what we say, actinostele. So what is actinostele? That is, xylem is a star-shaped. Okay. And what is third? Third we call plectostele. That means xylem is plate-like. Okay. So that is the protostele. That entire thing is protostele. Now, what is syphnostele? In case of syphnostele, what happens? Pith is present. Okay. So we can draw the diagram like this. Suppose this is a structure. So, okay, so let it be. This is a pith. So this structure, let it be, it is a xylem. Okay, and outer structure is a phloem. So this is a cyphnostele, right? So that is the only thing what we need to understand, that in case of cyphnostele, pith is present. Now solenostele, what happens? It is the most advanced. Or we can say found in fern. You know, fern is a higher pteridophyte, right? Or here what happens, that leaf cap is there. Leaf cap is present. Okay. So write on this. Done.
Done, sir.
Okay. So now we will talk about, that is reproduction. Okay. So, here what happens, that first of all, we need to understand that sporophyte bears sporangia. Sporangia, right? Which contains spore mother cell. Obviously, it is a diploid. Now, what happens, that spore mother cell undergoes meiotic division to form haploid spore. Right? Now, spore after liberation germinates to form prothallus. So, minded, prothallus marks the beginning of gametophytic stage, right? So, we have always emphasized on that. Fungal spore germinate to form germ tube, bryophyte, protonema, pteridophyte, protothallus. Okay. So, what we can say, it is a gametophytic generation now. So, in case of gametophytic generation, what happens? Prothallus bears sex organ. Okay. It can be monoecious as well as dioecious. So you understand the meaning of monoecious and dioecious, that male and female in the same plant, in the same prothallus, or there are different prothallus. Okay. Now you see male sex organ, or rather we can say that or we can say that sex organs are embedded in the prothallus. Okay. So you see, male sex organs is known as antheridia. And female as archegonia. Okay. Right. Now let's talk about the archegonia first. Now you know, archegonia is a flask-shaped structure. So we can say archegonia is like this. Okay. And you know, it is going to be covered by multi-layered jacket cells, right? So this area is actually called as a neck. And we can draw like this, that is the multi-layered jacket cell is there. Okay, so these are the jacket cells. Okay, and there is a binucleate neck canal cell. So we can say it is a binucleate neck canal cell, and they have a one venter canal cell, right? And this is one egg, right? You know, this entire sex organs is embedded, but archegonia is embedded with slight projecting neck. So what we can say that archegonia is embedded with slight projecting neck. Okay. And one more thing, that number of archegonia varies from 3 to 6. You see, the number of archegonia from bryophytes onwards, it is decreasing. In case of bryophytes, the number of archegonia were much more numerous. But here, there you can see the tendency in the decrease in the number of archegonia. It has reduced from 3 It has reduced from 3 to 6 only, right? So just write on this.
Or at the end of the class, can you just give the ploidy of all the structures, because I'm not getting it anywhere in the textbook and I can't remember them all.
Ploidy of which structure?
All the structures for like in the angiosperm. So the megaspore mother cell is N.
Megaspore mother is always 2n.
Sorry, mother is 2N, which will be both will be female. And then the megaspore is N, which is only female. And then the endosperm is 3N, which is 2 female, 1 male, right? And in the gymnosperm, what is the for endosperm?
That is an haploid.
And that is again only female.
Correct.
Okay, so that is it. Now there's no more structures which they can ask for ploidy, right? In hybridization.
No.
Okay, sir. Thank you. Done, sir.
Done.
Yes, sir.
Okay. Right. So next talk about Anthridia. So Anthridia is a globular structure. Enclosed by multi-layered jacket cells. So Antheridia is going to produce a sperm, or what we call amphizoids. These are unicellular biflagellate, or it can be multiflagellate as well. Okay. So what we can say, it produces whenever it is unicellular, then it is a biflagellate. Biflagellate male gamete. Okay. And that is going to happen in case of Selaginella, that we observed in case of Selaginella. But in the most of case, mostly it is in the form of multiciliate, right? However, it is mostly multiciliate. Okay. Now you see the movement of male gamete is chemotactic. Movement of male gamete is chemotactic. Okay. Chemotactic, that means it is under the influence of some chemicals, right? So what is the name of the chemical? That is malic acid. So it is under influence of chemical known as malate. Okay. Mind it, in all the species, it is the malic acid that going to attract the male gamete, except bryophyte. You know, in case of bryophyte, the male gametes are attracted by sucrose. Okay. So that is about anthrozoids. Now you see, fertilization require moisture. Okay, hence it is also known as amphibian among the plant kingdom. Known as amphibians among the plant kingdom. Okay, quickly write down.
So in the first point, it says globular structure enclosed by, I can't read that word.
Enclosed by multi-layered jacket cells.
And in the third point, movement of the gamete is
Movement of gamete is chemotactic. Tactic is a word. We will discuss the word tactic. It is a technical term. We will discuss in the plant physiology. So just write down chemo, CHEMO, tactic.
Done, sir.
Okay. So we have just seen the entire general features of bryophyte, right? You know, we call bryophyte as a, a higher pteridophyte as a fern, right? So now what we will do, we will study about the fern. So fern is actually called as a higher pteridophyte, right? So first of all, let's try to understand that how actually it is placed. So we can draw the structure like this, that suppose uh, this is a, so this is a structure. So this is what we call rhizome, because we know very well stem is in the form of rhizome, right? And these are the adventitious root. Okay. Now from here, the stem arises, right? And so when the leaf is going to form, there is a very typical structure form. This is what we call circinate vernation. Okay. Then the leaf will form like this. Okay. So within that leaf, what we will see that at the end of midrib, the spores will be present. Okay. So this is actually called as a sorus. Now sorus bears sporangia. So if I enlarge this structure, we can draw a structure like this. So what happens? This particular structure is actually called as an indusium. That is a productive structure. And what is the function of indusium? That it protects sporangia, right? And from here, we can say that large number of sporangia is actually formed. So that is the sporangia, right? So this is entire structure is of sorus. Within sorus, this is the sporangia. Okay. So quickly draw this diagram, then we will write on the entire points.
Done, sir.
Noted? Right. So again, we will just note down the important points. The first being that leaf bears spores and known as a sporophyll. Sorus is present. At the end of vein of leaf. Each sorus contains sporangia. Okay, write on these 3 points.
Done, sir.
Okay. Right. So what we will see that what are the exclusive features of the fern? So these characters which we are going to discuss, these are not found in other pteridophyte. Okay, the first, that is presence of hair-like structure called ramenta. So that is found only in case of fern. Number 2, heart-shaped prothallus. In lower pteridophyte, what happens? Prothallus is of any shape, thallus-like. It is also thallus-like, but the shape of the thallus is heart-shaped. That is in case of fern. Okay. Third, leptosporangiate. And lower pteridophytes are eusporangiate. So what is the meaning of leptosporangiate? That single cell form the entire,
What is the hair-like structure called?
sorry,
What is the hair-like structure called?
ramenta, R-A-M-E-N-T-A.
Okay.
Leptosporangiate, single cell form the entire spore, right? Okay. Now, what is the fourth? Presence of inducium, right? So this is the fourth character. And the fifth one, which I have shown you, that is circinate venation. That is coiling of the leaf before the opening. This is what we call circinate venation. Okay. So these are the 5 characters exclusively found in ferns. Okay.
So what is the fourth point?
Presence of inducium.
What is inducium? Sorry, I missed that.
We have drawn in the diagram now the structure.
Right. Sorry, okay. Done, sir.
Okay. Now let's discuss one last thing, and that is seed habit. Seed habit of Selaginella. First of all, in pteridophyte, seeds are not formed. Okay. If I'm saying seed habit, that means there is a tendency towards the formation of seed, right? So seed is not formed. So this has been observed in Selaginella. So what are the requirement for the seed habit? The requirement is the plant should be heterosporous. Okay. So presence of two types of spores. Okay. And what we call them? Heterosporous. Okay. These are two types of spores, right? What are the two types? One, what we call microspore. Second, what we call megaspore. Okay. But mind wait, pteridophytes are basically homosporous. So you just write on this somewhere in the bracket that pteridophytes are homosporous. That means they have only single type of spores. Okay. Or we can say only 8 pteridophytes have heterospory. Selaginella is one of them. Okay. Then second, what we can say, that retention of the megaspore. Megaspores are not released, right? So retention of megaspore by plant. So that is also the seed habit. The number 3, what we can say, that after fertilization, the entire thing is protected. So that gives some sort of sense of the seed coat. Okay. So what we can say, that presence of strobili, right? And fertilized structure is protected by covering, which shows tendency towards formation of seed coat. Tendency towards formation of seed coat. So we can say that seed as well as seed coat, both tendency is there. But once again, I would like to reiterate, no seed is formed. Seed is observed in gymnosperm. In angiosperm, no seed is formed. So there is only tendency is found. That's why we call them the seed habit of Selaginella. Did you get it? Understood? समझा क्या?
Yes, sir. Done, sir.
डन। चलो फिर एक और लास्ट चीज बताता हूँ। एक प्लांट होता है जिसको हम लोग कहते हैं रिसरेक्शन प्लांट। तो आपने देखा होगा एक्वेरियम में जो कुछ प्लांट होते हैं बिल्कुल ग्रीन नजर आते हैं पानी में, राइट? लेकिन पानी से उसको बाहर निकालो, दे बिकम अ ड्राई और ड्राई ब्राउनिश कलर। So these are called resurrection plants, which actually they become green in the plant, green in a water. So one of the important resurrection plant is Selaginella lepidophylla. So that is widely used in aquarium, right? So just for a reference, you can also write down that it remains green inside water. Okay.